Our final speaker in this session is Jaideep Mavoori, who has over 15 years of experience on semiconductor engineering. He has over 12 publications, is currently the senior research scientist at NeuroVista, where he develops medical implants for epilepsy management. Dr. Mavoori, please. That advances the slides at this point.
Well, thank you, Dr. Plumber. And thank you, Dr. Rothblatt. It's an honor and a pleasure to be here. It's been a great set of speakers and presentations. But in this talk, I would like to share with you some excerpts from my research with miniature animal-computer interfaces. This was done at the University of Washington with my very brilliant collaborators, Andy Jackson, Eb Fetz, Tom Daniel, and Chris Terrio. I should confess, when we started this research, telemedicine was but a little speck on our radars. Instead, our objective at the time was to shed more light on the brain-muscle puzzle. By that I mean all the intriguing questions about how is it that we perform the complex maneuvers with all the limbs, as well as our finger control, as well as motor learning. And rather than using the conventional techniques that neurophysiologists use, where you train animals to perform repetitious tasks, and while the animals are performing them, you record signals from the brains and muscles, and you look for patterns and correlations within the data and try to draw inferences to what the underlying neuromuscular mechanisms might be. We thought we would have a better go at solving this brain-muscle puzzle by looking at the same biological signals in naturally behaving animals. We felt that learning would be very valuable for designing advanced neural prosthetics. And it turns out that it's quite valuable for neural telemedicine as well, which I hope to show you today. I've sketched some basic blocks for what a telemedical, or specifically neural telemedical systems functions ought to be. You need functions to acquire and monitor the neuromuscular signals. These could be at a variety of spatial scales. You could get data from single neurons, from local field potentials, from small pools of neurons, electrocardiograms from even larger populations of neurons, as well as electroencephalograms that you can get non-invasively from the scalp, and electromyograms from your muscles. The system should then be able to detect abnormalities and even track their pathological evolution. And it could then transmit the data to a remote physician, or if you can embed the algorithms into the system, it can diagnose what the ailment might be and issue alerts as appropriate. And if it is suitably plumbed and has the mechanics, it can initiate certain curative actions. For example, it can stimulate electrically to induce any long-term biological repair mechanisms. And you'll see some of those examples with neural plasticity in this talk. Or it could do short-term symptom suppression via a drug delivery or other focused electrical stimulation. Well, the neurochips that I'm about to describe and the experimental results map quite nicely to each of these functional blocks. Let me give you some more background. One of the animal models we used for studying this biological motor control problem is a macaque. Its neuroanatomy is very similar to that of humans, and hence a very good translational research subjects. The pumpkin, if you're wondering, was because this picture was taken during Halloween time, and the monkeys were playing with pumpkins, yeah. Now, if you were to walk into a neurophysiology lab, you would expect to see instruments such as these. And these are bulky, non-portable, certainly not for the monkeys. And because of the size constraints, the monkeys are placed in the vicinity of these instruments. You have wires connecting to their neuromuscular tissues. You need functions such as filters and amplifiers to precondition the data, analog to digital converters to digitize, and hard disks to store your experimental results, spike discriminators to extract the neural spike shapes, and stimulators to perform stimulus response experiments on the animals. Now, as I mentioned, we wanted to do these experiments in naturally behaving animals, so we had to compact all this setup. There are different ways of going about it. One, you build a transceiver, mount it on top of the monkey, and as the system collects the data, you, in real time, transmit it to a remote station where you're doing all your analysis, your spike extraction, and sending commands back. These could be to stimulate or to alter the course of your experiments. But such a system would consume a lot of power because radiotelemetry takes power, as you heard from several speakers here. It limits the range of separation between your animal and your analysis station. And if you want to do any sort of real-time, tightly coupled closed-loop experiments, your transmission latencies are going to come in your way. So there's another option. You take all your analysis instrumentation, compact it, and put it on the monkey. Such a system will have to operate autonomously. It's got to be able to crunch the data, locally compute the experimental data, make decisions on the basis of the data. But because you're not transmitting data back and forth, it could consume low power. Also, you might wonder, if you're compacting all this, you might lose some processing capabilities. But with some clever algorithms and techniques, you can still perform some very valuable experiments, as you'll see. Our own approach was a hybrid between the two. We did have a wireless interface, but rather than using it for real-time communication, we would use that to download. This was an optical interface. So we would download experimental parameters at the start of an experiment, have the system perform a sequence of experiments and store its experimental results into onboard flash memory. And at the end of the experiment, we would extract the data for further analysis. Here's how our system looked like. We took the essence of the functionality, compacted them into circuits that would fit into a 60 millimeter diameter titanium casing. You can see the optical interface there. The battery powers the system, depending on the nature of the experiment you're doing, upwards of a day. And the system gets its neuromuscular inputs via these connectors. The orange block, as you see in this cartoon here, has wires leading into a microwire array that's implanted into the primary motor cortex in a region that was responsible for the forearm wrist muscle control. And the white connectors lead to wires that were percutaneously tunneled and implanted into the contralateral forearm muscles. You can see a cartoon of how the system is mounted on a monkey, and it extracts data while the monkey is naturally behaving, foraging for food, playing with toys, and other interactions. You can, I did not see Dr. Bruce's talk or any of the other talks, but we do have a palm interface to talk to the chip. We have some crude analysis routines for that. But more typically, we tend to use a PC with an optical interface, and we have a suite of analysis routines that lets us extract and analyze the data that's been collected by the neurochips. Let's look at the architecture a little bit. It's a dual process system. There are two mid-signal processors, one for neural signal analysis and another for muscle signal analysis. They talk over an onboard network. The system is scalable. You can add additional processors for adding extra channels or increasing the computational resources on your system. The neural signal processor takes data from one of the microwires within that orange block, filters, preamplifies, digitizes it, and the discriminator extracts the neural spike shapes. And the stimulator module delivers high voltage but current limited pulses via another microwire. I'd like to draw your attention to this loop here where the signal extracts the data, extracts the spike shapes, and conditionally stimulates via another microwire. We were using that to create artificial recurrent connections within the brain to induce plasticity and to remap some functionality there. The EMG, or the muscle processor, takes data from its own microwires, rectifies and averages it optionally to give you an aggregate muscle activity. Okay? And the two of them operate in parallel, synchronously, so they can time sync both their operations and their statistical computations. let me show you some data clips collected by the neurochip. Here's a 1-second clip. The top plot shows the cortical activity. You can see the spikes. When you insert a microwire in the cortex, you tend to pick up activity from any number of surrounding neurons. But in any experiment, you may be interested in activity of just one of those neurons. But luckily, each neuron tends to have its own unique shape. And so if you program the shape parameters for that neural action potential, you can have the chip scan the activity in real time, and whenever it encounters an action potential of a particular shape, it catalogs it. So in this example, you can see, there are two different neurons spiking in there, and the neurochip in real time has identified for the neuron for which we have programmed the shape parameters in red. And here's an overlay of all the spikes. This is a zoomed-in overlay of all the spikes. And they fall into two categories, the black ones and the red ones. And the red ones do overlay quite nicely, indicating that it was the same neuron that the chip was tracking through this 1-second period. You can do this for much longer times, as I'll show you. These bottom plots show activities from two muscles, an extensor carpi radialis and a flexor carpi radialis for the wrist muscles. And these are rectified activity there. As I mentioned, the neurochip can catalog the activity. So you can program it to count the number of spikes occurring in, say, 100 millisecond interval. And from that, you can compute the firing rate. So here's a segment done over 1 minute. And you can see the corresponding muscle activity, the average muscle activity. I mean, you can visually see some of the correlations within this data, the swings in the firing rate and the swings in your muscle activity there. And you can do this over 12 hours as well. We believe we will be one of the first to collect this data in a naturally behaving animal. You can see during daytime, when the monkey is busy with its tasks, the cortical activity out of this one neuron is elevated. But at night, when the monkey goes to sleep, the neuron enters a slow wave bursting activity. These are coincident with the REM cycles. The muscle activity correspondingly is high during daytime, and at night it is suppressed. And this occurs in all of us. Our brains inhibit muscle activity or the descending motor commands at nights, so you don't end up sleepwalking or enacting your dreams. So here's 2 weeks in the life of a single neuron. The spike shapes overlay quite nicely on each day, as well as from day to day. And you can also see in the blue plot here the daytime activity and the nighttime cycles. And as the monkey wakes up, the neural activity starts picking up. You can see the pattern repeated from day to day. Once you start collecting this data, with all these new capabilities of the neurochip, you can start asking questions about what the underlying system properties might be. And here are some questions we were interested in. Questions such as how time invariant is this neuromuscular system? How does the model compare during task and during free behavior? And chief in our minds was, can we alter the system's properties? I'll give you some samplings of experimental results to answer each of these questions. Do we have 5 more minutes?
You have 1 minute.
I'm sorry?
Yes.
Yeah, 15 minutes. Okay. Okay. So here's data collected over a 4-hour duration. The top one shows the firing rate from one of the cortical neurons and muscle activity from 2 antagonistic muscles, again, an extensor and a flexor, an extensor carpi ulnaris in this case, and a flexor carpi radialis. During the first 20 minutes, the monkey was doing a trained task. It was seated in front of a computer screen, tracking cursors over different trials, with its arm strapped in a joystick or a manipulandum that was also measuring the torques exerted by the wrist muscles. And during this time, the neurochip was collecting data from its brain and muscles. During the remaining 4 hours or so, the monkey was let loose. It was, again, indulged in its natural activities. And during the whole time, the neurochip was collecting data. And after the end of that session, we extracted the data for further analysis. We did this over several muscles, several neurons, in multiple subjects as well. But you can analyze this data at a multitude of time scales. Here, for example, by zooming in on the task data, you can see the trial-by-trial modulation of the cortical activity as well as the almost antagonistic activity from these two muscles there. Let me give you a summary of these findings rather than going to all the details. We found that over several neurons and muscles, aspects of the system are time invariant, and they're pretty linear too. The second thing was the relationships translate for the most part from task to free behavior, and interestingly from day to day as well. So as you can imagine, these findings are very advantageous for neural prosthetics because they imply that a quadriplegic or a paralyzed patient wearing one of these arms can train the mechanical parameters for his arm during a training task and have it help him through the day-to-day natural activities. Let me jump to the a second set of experiments where we were trying to alter the system's properties. These experiments relate back to the Hebbian postulate, which simplified reads neurons that fire together should wire together. So if you recall, I pointed that loop in the architecture diagram. So we created this artificial recurrent connection. We took one of the electrodes called the recording electrode and a stimulating electrode, and we extracted action potentials from the recording electrode, had the neurochip trigger a pulse or deliver a stimulus through another microwire. So we created an artificial synchrony between two pools of neurons within the brain in an area responsible for the wrist muscles. And the question was, does this induced correlated firing create long-lasting changes in the biological wiring? And we found across multiple neurons, multiple muscles, multiple animal subjects that is true. We do cause changes in the biological wiring. Here are details from one of these experiments. Oh, by the way, several people have done these experiments with slice preparations, but in vivo evidence for that has been lacking. And I mean, until these experiments came along. So here are details from one of the experiments. Again, we took 3 electrodes. One was a recording, another was a stimulation electrode, and a control electrode. Now, at the start of the experiment, we characterized the stimulus response from each of these microwires. The recording electrode recruited activity mostly in the flexor carpi radialis, and the stimulation electrode in the extensor carpi radialis, and the control electrode in the flexor carpi ulnaris. And then we hooked up this recurrent connection using the neurochip, let it operate for 2 days in this case, but normally it was 1 day. And then we recharacterized or tested the system, the stimulus response from each of these electrodes. What we found was the control and the stimulation electrodes remain unchanged, but the activity from the recording electrode, it recruits activity in the flexor carpi radialis just as before, but it also activates the extensor carpi radialis. So we created mapping between the flexor and the extensor muscles in the system. So the movements evoked from the recording site are changed to resemble those evoked from the stimulation site. Again, here's a summary of the findings. We found that the timing from your spike, from the action potential to the stimulations is critical. But 20 milliseconds is a sweet spot. Anything below or more decreases the strengthening effect. So there is a spike timing-dependent plasticity component. Also, even though you're conditioning the system for just about a day, the effects themselves last for a much longer time, over 10 days in some cases. So you have actually reprogrammed or you've strengthened the memory of the connections between your brain and your muscles. So again, this could be very useful for repairing damage caused by spinal cord injuries or by other neural disorders. So I guess to summarize what you've seen so far, we saw that the neurochip can monitor activity. It can track the activity of the cells and can automatically determine correlations in your system. Functions that could be used for detecting early onsets of neural disorders and diagnosing ailments in your system. And it can also repair in terms of remapping if it senses there's a weakening of say, some neural or muscle connections. So I do hope that even though we might be at the early stages of neural telemedicine today, we would leapfrog and maybe use systems like these implantable computers to be able to detect early stages of neural disorders and prolong the fruitful duration of our lives. I wanted to end with one other slide to expose you to the engineering possibilities here. So we've taken some of the neurochips, the designs, and we have shrunk them over several generations to perform or to study this biological motor control problem in flying insects. So here's one of the prototypes here. It's small, has very similar functionality of the stimulation, recording, storing results on board, and having a wireless interface. And these insects are giant hawk moths, Mundicus sextus, and they can carry these chips comfortably, and you can perform experiments as they're flying around. And wish to close by acknowledging my collaborators and funding from NIH, Office of Naval Research, UW Royalty Research Fund, and the Packard Foundation. Thank you.